Hi guys, so for today's experiment, we will be doing biological molecule. So first, let me introduce to you the materials. First, we have an orange here for our enzyme. Next, for our protein, we have an egg. And next, for our lipids, we have an olive oil. And for our carbohydrates, we have bread here. And also, we have scoops and strainer here that we will be using also. After we completed the experiment, we'll need to answer the following questions. Number 1, what happens if we submerge the protein in the enzyme? Number 2, what happens if we submerge the lipid in the enzyme? Number 3, what happens if we submerge the carbohydrate in the enzyme? And number 4, what changes do you notice in the 3 specimens after 30 minutes and an hour of submersion?
Our hypotheses are: first, if we submerge the white egg protein in orange juice, the enzyme, after 30 minutes and an hour, then it will gradually turn into a visible white color. Second, if we submerge the olive oil, which is lipids, in the orange juice, which is enzyme, after 30 minutes and an hour, then it will only place at the top of the enzyme because it is a lower mass than the orange juice. Lastly, if we submerge the bread, the carbohydrate, in orange juice, the enzyme, after 30 minutes and an hour, then it will absorb the enzyme, causing its size to gradually increase.
The first specimen is white egg. Before, it looks like a crystal clear. The second specimen is the olive oil. Before, it is clear and normal. And the third specimen is bread. Before, the bread was dry and of a small size. After 30 minutes, the white egg's side became visible. While the olive oil, nothing happened. It is still clear and normal at the top of the enzyme. The bread, on the other hand, becomes wet because the enzyme is absorbed by the bread. After an hour, almost all of the white egg has turned to a visible white. And still, nothing had happened to the olive oil. The bread, on the other hand, is completely wet as a result of the enzyme being absorbed, causing the bread size to increase.
In conclusion, our hypotheses for this experiment were all correct because first, after the white egg or protein was submerged in the enzyme for 30 minutes and an hour, it gradually turned into a visible white color. The second is that after 30 minutes and an hour of submerging the olive oil or lipid, it did not mix with the enzyme. Instead, it only placed into the top. Lastly, after submerging the bread or carbohydrate in the enzyme for 30 minutes to an hour, it completely absorbed the enzyme, causing the bread to slowly inflate.
So, as the specimens are all submerged in the enzyme, different changes in each specimen can be observed. The white egg gradually turns into a visible white color after 30 minutes and an hour of being submerged in orange juice. The olive oil, on the other hand, only plays on top of the enzyme because it has a lower mass than orange juice. Moreover, the bread or carbohydrate absorbs the enzyme, causing its size to gradually increase.